Hey guys, welcome back. We're going to move on with 8.4 here. We're talking DNA in AP biology right now. So let's jump back into it. Eukaryotic chromosomes. So eukaryotes, we're talking things with membrane-bound organelles, nuclei, you, me, plants, dogs, cows, fungus. You get the idea. So DNA in eukaryotes, it's in the nucleus, most of it. It's organized as one or more chromosomes that differ in length and shape. So you've probably heard that humans have 23 pairs of chromosomes or 46 chromosomes, something like that. So a chromosome is a structure that consists of DNA and associated proteins. DNA is super, super long, but we have to fit it in a very small space. The nucleus. So the way we do that is we wrap it around these proteins called histones, and then we kind of braid these proteins together and we get this really, really dense, tightly compacted structure of DNA. And we can end up getting like almost 3 feet of DNA or something ridiculous like that, packed into this very, very tiny nucleus that you can barely see under a microscope. So very cool. The chromosome is going to carry part of a cell's genetic information. So, how they're organized during most of the cell's life, your chromosome really consists of one DNA strand. It still looks like a chromosome. It's just one copy of that chromosome because that's all you need to go through your day-to-day life. Now, when your cell wants to divide, we're talking mitosis, maybe meiosis, it's going to duplicate all of the chromosomes so that both of the offspring, which could be offspring cells, like just daughter cells, the cell's just going to pinch itself in half, or this could be an offspring like we're going through meiosis and truly making sex cells. So either way, you want both offspring to receive a full set of chromosomes. You want each new cell has one copy of chromosome 1, one copy of chromosome 2, etc., etc. You don't want any information left out. So when you get a duplicated chromosome, you get two DNA strands. And once we've gone through this duplication process, we don't call these guys chromosomes anymore. We call them sister chromatids. So they are copies of the chromosome. We have two now. And they're attached to one another at a structure called the centromere. And that's important. So they consist of two really long filaments. And when you get these two long filaments that are tied together by a centromere, most of your chromosomes end up looking kind of like an X. So here you have an unduplicated chromosome, just a cell being a cell. Comes time to divide, it copies all of its information and makes an exact copy of itself. And they are pinched together here in the centromere by a kinetochore protein, little bonus information for you there, with one sister chromatid on top here and one sister chromatid right here. So that's duplicated chromosome. So our chromosome consists of filaments, and that's just the term for the little blue line you saw on the last page. And it's a coil of DNA wrapped around these spools of proteins that I mentioned before that are called histones. Those guys are very, very important. Allows us to get so much protein in such a small space, rather, so much DNA in such a small space. So each DNA histone spool is called a nucleosome. That's what we call if we just wrap DNA around one histone a few times, that's called a nucleosome. And that is about our smallest unit of how you can organize a chromosome in eukaryotes. In prokaryotes, the DNA is just kind of hanging out in there, all bunched up in a pile. So in eukaryotes, like you and me, we get nucleosomes and these basic units of chromosome organization. So the DNA molecule is going to consist of two strands, and it's twisted into a double helix like you've seen before. And so here's kind of putting together what we talked about last time with what we're talking about this time. It's a lot to take in, but here we are. One little piece at a time. Here's our nitrogenous bases. Here they're wrapped into a double helix. The DNA starts to wrap around these little purple balls we call the histone protein. They're not actually a perfect circle like that, but we'll go with it. The histone proteins start to hook together here with number 3, and they kind of twist themselves into a fiber. You see how this little histone protein has started to coil itself up like a spring? which is a little bit ironic because this guy was coiled up like a spring, but we'll go with it. So we've got a spring within a spring right now, and then to further complicate matters, we then take the spring that's coiled into a spring and we use it to make another spring. So we have 3 springs made of other springs, and then we coil it all together into this huge structure, which isn't actually that huge. It just looks like it in this picture, that we call a chromosome. And then here is the cell membrane of a cell, the nucleus, the nuclear envelope, and there are many different chromosomes, different lengths and structure. So one sister chromatid, one sister chromatid, one duplicated chromosome, ready for reproduction. Very, very, very cool. That's a chromosome. Here's some pictures of them. It's kind of cool. A little electron micrographs of chromosomes. Doesn't really look like anything, but there you have it. And then this guy, look at that. Beautiful. Screensaver quality shot. And then you have whatever this is. This is actually a little bit cooler, I guess. Super blue, fuzzy chromosome. One sister chromatid over here, one sister chromatid over there. 2 micrometers, so they're pretty far zoomed in. But who would have known that all the information that makes you look like you and me look like me would be a little blue fuzzy caterpillar. And of course, you know that color is added artificially. Electron micrographs don't give color images. So, the total number of chromosomes in a eukaryotic cell is characteristic of the species. Humans, we got 46. Corn and cows have like way more than us. Dogs, 72. They almost have double the chromosomes we do. I don't really get that, but there you have it. 2 of each type of chromosome, so their chromosome number is diploid. That's for us. Don't apply that to all sorts of plants necessarily, because plants can be pretty flexible. But for humans, 46. So we have 23 pairs. We're 2N. We have 2 copies of chromosome 1. You have 2 copies of chromosome 2, you have 2 copies of chromosome 3, on and on and on and on, until you get to 22. And then if you are a female, you most likely have 2 X chromosomes, and if you are a male, you most likely have 1 X chromosome and 1 Y chromosome. There are variations in that, but they are pretty rare. So a karyotype shows how many chromosomes are in the individual cell, and it reveals abnormalities if they're really blatantly obvious. All a karyotype is a picture. It's a picture of your chromosomes. And we take all of your chromosomes and we lay them out by size, smallest to biggest. And we take a picture of it. And that's a karyotype. And so here you have a karyotype of a human. There's 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3, and 4, and 5, and 6, and you can see they get progressively smaller till you get all the way down to X chromosome. Y chromosome is absent, so this would be what gender? Of course, it would be a female. And so what you can do here is you can look for things like, do they have extra chromosomes around? Do they have one chromosome that is significantly shorter than another chromosome? Are they missing genes? Are chromosomes broken? Things like that. That's what a karyotype allows you to do. And so that can be kind of cool. So autosomes as opposed to sex chromosomes, we've already kind of alluded to this because I said you had 1 through 22 and then X's and Y's. So in a diploid organism, like you and I, 1 chromosome in a pair is inherited from the mother and 1 from the father. So if we run back to this one, you could say, I got 1 copy of chromosome 1 from my mom, I got 1 copy of chromosome 1 from my dad. And on and on and on and on we go. 1 copy of chromosome 4 from mom, 1 copy of chromosome 4 from dad. Okay, the only thing I can't say that on is this one, depending on my gender. Because if you got a Y chromosome, you most certainly got that from your dad. Okay, this happens to be a female, so 1 X chromosome from mom and 1 X chromosome from dad. And that's where you get your DNA from. So, all except for 1 pair of chromosomes are going to be autosomes. Autosomes are body chromosomes that don't determine the gender, okay? So chromosomes, they have about the same length, they have about the same shape, they have about the same centromere location, and they are going to determine everything about you except for gender and a few other traits that are what we call sex linked. But we'll get to that later in this book, so don't worry too much about that yet. So pairs of sex chromosomes differ between males and females. Human females usually have two X chromosomes. Human males usually have an X and a Y. That's not always the case. Some animals have a lot more than just two chromosomes that determine gender. Some animals it's flipped, and the chromosomes that are different, like this one, make the female and this one make the male. But in humans, 2X make the female, X and the Y make the male. But know that that's not the case for all animals. Cool? Just us. Some animals, it can be the temperature can change their gender. I mean, weird stuff like that. So bear that in mind. So DNA replication, when it comes time to replicate DNA, to make a new cell, to make sex cells, whatever the case may be, DNA replication takes a lot of energy because there are a lot of enzymes working in this DNA. But what we're going to do is copy the DNA. And we have to be very careful about this because errors in this can be fatal. I mean, it's a big deal to mess up DNA. So a cell's going to copy all of its DNA before it reproduces, and each of the two strands in the DNA double helix is replicated. So we call this semi-conservative replication. DNA replication requires a lot of enzymes, like I mentioned before, including the heavy hitter DNA polymerase. It gets a lot of the attention because he's the one who actually attaches so many nitrogenous bases. But there are a lot of others that I want you to know. So when we go to replicate our DNA, the cell's genetic information consists of basically the order of our nucleotide bases: A, T, C, G, C, C, A, T, T, G, C, that kind of stuff. That's information, and that information is all contained in our chromosomes. So our descendant cells, whether you're making more hair cells or skin cells or liver cells or sperm or egg, if it's for reproductive cells, they've got to get the exact copy. They can't get deviations in this information because that can be really a bad deal. Now, sometimes that happens. Sometimes it's not such a bad deal. But we want to get an exact copy if we can. So each chromosome is copied in its entirety, and the two chromosomes that result are duplicates of that parent molecule. So it's an exact copy. So here's a few of the enzymes you want to be familiar with. DNA helicase breaks the hydrogen bonds between DNA strands. Now I really like this name because enzymes are generally named after what they break down or work on, and I think that's really cool. So for example, let's think of one. Lactase breaks down lactose sugar. Cool. So a helicase breaks down the helix of DNA. And so that enzyme breaks these hydrogen bonds between DNA nitrogenous bases, and it will actually unwind that DNA molecule. So it breaks the bonds, and then along comes another one called topoisomerase. And topoisomerase is going to be the one who untwists that double helix and makes sure it doesn't try to zip back up, so to speak, if you think about it like a zipper, with all the nitrogenous bases being the teeth in that zipper. Topoisomerase ensures that DNA doesn't hook back together after helicase breaks it up. So DNA polymerase then joins our free nucleotides into a new strand of DNA, and that's why he gets so much attention, because his job is super important. And then at the very end, DNA ligase goes and joins DNA segments onto this discontinuous strand. And we'll see a picture of that discontinuous strand here in just a little bit. So those are your enzymes that I want you to know about. So there's lots of different types of DNA polymerase. There's not just one who does everything. There's lots of different ones, and each requires a primer to initiate the process of actually making more DNA. So a primer is this short, single-stranded DNA or RNA that's complementary to a targeted DNA sequence. So if your targeted DNA sequence that you want to start at is, I don't know, AAC, okay, the primer you would build to that would be TT. TG. Cool? So the primer is the complement to the target. So the target is, I think I said AAC, right? The A bonds to T, C bonds to G, so your primer would need to be TTG. And then it's going to find that AAC in the DNA, hook in there, and serve as the primer. And DNA polymerase can grab that primer and start reading from there and making that copy. So that's what a primer is. It's very cool. So each strand of our DNA double helix is a template for a synthesis of a complementary strand of DNA, which basically means if you take that parent DNA molecule and you unzip it, you have two ends. Then if you copy those two ends, you have two new molecules, and each has half of that new strand is made of the original parent. And so that is semi-conservative. We haven't conserved all the DNA, we've conserved half of it each time exactly. And so one template builds DNA continuous, the other builds it discontinuously in little segments, and each new DNA molecule is going to have one new strand and one old strand. Now the reason we build them discontinuously on the second strand is because we have to read DNA polymerase only reads 5' to 3'. And so for one strand, because we mentioned earlier DNA strands travel different directions, for one strand he can just run up the strand. 5' to 3', just head up. As all these other enzymes open DNA up, he just follows behind them. The other strand, he has to try to run down the DNA strand, but it's not opened up yet. So he ends up building in these little tiny chunks from 5' to 3'. Still gets the job done, it's just not as fast. So here's what this would look like. Here's the DNA. We want to replicate it. So topoisomerase jumps on here, untwists that double helix. Helicase gets in here, breaks these hydrogen bonds, and we now have the parent strand, the parent strand. Cool? So that's the beginning. Enzymes unwind the strand. Primers, that's this little guy right here. These little primers serve as the initiation site for DNA synthesis. Starting at the primers, this big guy called DNA polymerase will assemble this new strand, which is in pink here, using the parent strand as the copy. So DNA ligase comes along, this little yellow guy here, and he's basically proofreading. He comes through and makes sure everybody's where they're supposed to be. A bonded to T, cool, let's move on. C bonded to G, cool, let's move on. He's making sure there's no mistakes, because if we make a mistake in here, this could be a really, really bad thing. So then we get each parental strand, these blue strands, serving as the template for the assembly of the new strand of DNA. Both strands of the double helix are going to serve as the template, and so two double-stranded DNA molecules result. One strand is parental or old, the other is new, and that's where we get this term semi-conservation. And so semi-conservative replication, we've got a blue strand with a pink strand, an old blue strand with a new pink strand. And that is how we take one DNA molecule and we turn it into two with identical information. We hope. Hopefully, identical information. This DNA polymerase, this is a 5' end, that's a 3' end, and he just goes trucking up here. Lots of DNA. Quick. Real quick. This guy, this is the 5' end, and this is a 3' end. So he jumps in, finishes a little chunk, breaks off. There's a new primer. Jumps in, finishes a little chunk, breaks off. And he's waiting for helicase to open up more 5' areas with primers. And so a little different way that these two guys get the job done. Same job, just a different way to do it because they have to travel in one direction. So discontinuous replication. This slide's going to show you what I basically showed you on the little cartoon in the last one. DNA polymerase attaches free nucleotides only at the 3' end. Only one of the two of the new strands can be synthesized continuously. Synthesis of the other strand occurs in little segments, and DNA ligase joins these guys into one continuous strand when it's all done. So there's the picture of discontinuous 5' to 3', 5' to 3', a DNA ligase is going to come in, make sure these guys are all patched up. And right here you have the adenine that wants to bond to the thymine. So as he comes in here, the OH group is going to bond to his phosphate. He'll lose phosphates. It gives off a lot of energy. Actually provides the energy to form this bond in the first place. It comes from these phosphates. Does it remind you of ATP? Because it should. Adenine bonds to thymine, and we're in good shape. On and on and on it goes. It's crazy. That's the information in your body. Weird. So weird. So, mutations. Everybody knows about mutations. We learn about them when we're little kids when we watch the X-Men, right? People get wings, and they can change the color of themselves, and they get all these really cool skills like moving metals with their mind. And then you take biology with me, and I crush all your dreams and tell you that Spider-Man's not true and it's depressing. But really, mutations stink. Yeah, they're a bad thing. DNA polymerase proofreads the DNA, right? Ligase, making sure the DNA is cool. If there's damaged DNA, we're going to take it out and try to repair that. But when we can't, or these mechanisms fail, or we're continually exposing ourselves to chemicals that cause DNA mutations, an error becomes a mutation. And all a mutation is a permanent change in the DNA sequence. And that is probably not good. There's a lot of different things that can give you mutations. Radiation, ionizing radiation, gamma rays, think Incredible Hulk, right? Doesn't actually end that way. X-rays, UV light, these guys are so powerful that they can knock electrons out of atoms, and they can over time break chromosomes, and little pieces of the chromosome can get lost. Other things these guys can do is create free radicals, and free radicals are a atom with an unpaired electron, and it wants to fill that octet so bad it will go and rip electrons from whoever, wherever, and now you have unstable molecules in your body. And if that's in your DNA, you could be in bad shape. So UV light, if it's between the range of 320 to 400 nanometers, it will form these little pyrimidine dimers that kink the DNA strands. Your book has a really cool picture of basically two thymines that are next to each other, that instead of bonding to adenine, they've actually bonded to their neighbor above and below. And it makes this weird little kink in the DNA strand, and that will lead to the formation of skin cancer. And so we have an actual biological reason that long-term exposure to UV light, or perhaps even short-term exposure of really strong UV light, can lead to skin cancer. So chemical agents. It's not just sunlight and gamma radiation. 55 carcinogens, carcinogenic compounds means they're clinically shown to cause cancer, are in tobacco smoke. Now, it's cool that we live in a generation where smoking is like not really cool anymore, but that used to be something that people thought was cool, and they will transfer these little hydrocarbons groups to your nucleotide bases in your DNA, and that will cause mutations in your DNA. And then you're on that long, painful road to the 85% mortality rate that is lung cancer and a whole host of other cancers. So that's not fun. Do me a solid, don't smoke, okay? We've got big enough things to worry about in our world. Don't smoke. Many environmental pollutants, of course, can also be converted by the body to other compounds that bind irreversibly to DNA, and once they're bound to that DNA, they cause replication errors. Those errors lead to mutations, and on down the road we go to just messing up our DNA. Not good. So, here's a case in point. I told you Rosalind Franklin was wicked good at taking x-ray pictures of crystals. Well, guess how she got that good? Practice, 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 right? So Rosalind Franklin spent a lot of time in the 40s in front of an x-ray without all the cool safety equipment that we have today. So, in science, public recognition does not always include everyone who contributed to a discovery. Rosalind Franklin, she took the picture that showed the molecular structure of DNA. Without her picture, Watson and Crick probably would have been building a long time, build models without getting anything right. But they took her picture, they got it right. Watson, Crick and Wilkins got the Nobel Prize. Rosalind Franklin got ovarian cancer and died at 37. Why? Probably because she spent a big piece of her life being exposed to x-rays for her entire working day. Okay? The Nobel Prize cannot be awarded posthumously. You can't get it if you're already dead. So the tough part about this is Wilkins worked down the hall at the same university as Franklin. They studied the same thing. Wilkins got her picture to these guys. He got the Nobel Prize. She died of ovarian cancer. Not a fun ending. But her picture is in your book, so I guess she's getting some credit now, which is good. But now we know X-rays, mutations, DNA. It's all together. Even in the discovery of DNA, it's all connected. So when we look at what we know about DNA, and we ask ourselves, should we be cloning animals? That's a tough question. All a clone is an exact copy of an individual. This actually occurs in nature all the time. Asexual reproduction. Lots of simple animals asexually reproduce. Lots of protists asexually reproduce. Or splitting an embryo. This happens naturally in humans. We call them identical twins or dizygotic twins, right? The embryo early in the stages splits into two. It has identical DNA. You end up getting identical twins. They look alike because they have the same exact DNA. As these cells start to develop, they become differentiated, which means they become different in their form and their function. Now, they do this by expressing some genes and turning other genes off, or parts of other genes off. And we use reproductive cloning technologies now to produce an exact copy or a clone of an organism. That's where we are now. And so we call this process somatic cell nuclear transfer, which basically means transferring the nucleus of a body cell. So we don't have to use sperm or egg to accomplish this from the donor, which is kind of cool. So we take our nuclear DNA of an adult, and let's say you've got like this cow that's won great, wonderful agricultural awards, and you'd like to win more great, wonderful agricultural awards. And so instead of going through all this process of trying to figure out how to breed a new calf that would make a good cow to show, you just take skin cells of your already award-winning cow and clone that cow because you know it will be good. You've already done it with this cow. So you take your nuclear DNA from this cow and you transfer it to an egg of a different cow that we've taken the nucleus out of. And you give them a little jolt of electricity, and that egg cytoplasm will reprogram that differentiated adult DNA to act like undifferentiated embryo DNA. And we call this a hybrid cell. This hybrid cell develops into an embryo that has the exact DNA of previous award-winning cow. And it's genetically identical to the donor. And here's what it looks like. You take your cow egg, you take this little glass suction tube, you pull this purple DNA out. We don't want that in there. Get out of there. He's out. You take a different micropipette, and you take the nucleus of your award-winning cow, and you inject it. So I have egg, cytoplasm, brand new nucleus. He's in the egg. We take it out. We have a little hole in our cytoplasm, which I might not be too happy about, but I'm sure it'll be okay. Give him a little jolt of electricity, and that cytoplasm's like, okay, nucleus, I guess you're mine now. And we go on to make a brand new copy of something that's already an adult. Very, very weird. And so, here's probably our last topic in the chapter is therapeutic cloning. What if you don't want to make an award-winning cow? What if you want to try to fight a disease? Can you use the same somatic cell nuclear transfer, cloning, to produce human embryos? What if we took one human embryo and a whole bunch of eggs, and we made thousands of human embryos, and then we had thousands of cells that were undifferentiated, and we could take these undifferentiated cells and do research with them from these cloned human embryos. And the research could ultimately lead to treatments for people who suffer from fatal diseases. Now, this is a hot button topic because human embryos, a lot of people consider that a life. And some folks consider it a blank slate of genetic information that's undifferentiated. And the way you view that and what actions we should take based on that is a big piece of what's called bioethics. It's how you make the decisions you make about science and biology in particular. And I'm not here to tell you there's a right or wrong about that. We'll talk about it in class. We'll kind of get some ideas from you guys, but it exists and it happens. And the research is interesting, but it comes at a cost. So whether the potential benefits outweigh the cost, that's what we decide when we vote on issues like this and when these issues come up in research. So that can be a tough thing to kind of talk about, but we're going to talk about it, so that'll be a good time. And I think that might be the end of your chapter, guys. I think that was about the last thing in there. So let's see. Yeah, that's it. Points to ponder. A few questions. One of the problems with cloned organisms, they tend to die pretty quick. Why do you think that is? We can talk about that in class. What are some reasons that DNA is double-stranded instead of single-stranded? Kind of a tough question, but we can talk about that as well. And then last one, what are some advantages of semi-conservative replication? Why is that a good thing? So I will leave you with those. I thank you for your time, and that's chapter 8. So thanks, guys.